All right, today we are going to do day 9 of STAAR Review. And the reason it's a video is because we're probably learning in place because there's like CSI testing and ACT going on for upperclassmen. The focus of today's review is on the cell cycle. So we already know that the cells in your body grow and divide throughout this cell cycle. And there's a diagram on the left side here that kind of shows you all the different phases. We'll talk about them all in detail and about which ones are the so-called important ones and which ones are pretty chill. Now, because I don't have you necessarily in my classroom, if you need to for the notes, you won't have a physical copy, but underneath the Pear Deck, there's another post that has the note sheet. You can open that one up and it's a color version, so it's pretty useful that way. So the first part that we're going to talk about is interphase. And interphase is like the majority of the cell cycle. It's like 90% of the cell cycle. And this is because 90% of the time your cells are growing and doing their job. And for a small part of that, they're also replicating their DNA. The other small phase within the cell cycle where the cell is dividing its nucleus is called mitosis. And at the end of mitosis, as you can see here in this diagram, there's this other phase called cytokinesis, where the cell's physically separating into two. And then both new cells will enter the G1 of interphase again. So there are three technical phases within interphase. First, we start out with G1, S, and then G2. Now the order does not go G1, G2 immediately. The S phase is sandwiched in between both growth phases. The G1 phase is the normal growth and function of the cell. It's growing in size because it has just finished splitting from mitosis and cytokinesis. So it's got to grow and it's got to do its job the whole time it's growing. Then it enters S phase here, found in the middle between the two G phases. And that's where DNA is synthesized. The S in DNA synthesis is the S for S phase. Here, the DNA is doubling. After the S phase, the cell will enter G2 phase, where the cell is then going to prepare to divide again. It should have grown enough. If it hasn't, it'll grow a little bit more. And it's going to double check that everything went well in the S phase, and we have all of our organelles doubled as well, so that when you do mitosis and cytokinesis, you have enough organelles for both cells. Here's a question for you to answer, just as a review of our last slide. It says, what occurs during the S phase? Is it A, DNA synthesis, B, DNA transcription, or C, DNA deletion? Go ahead and answer that in your Pear Deck. The correct answer here, obviously, is DNA synthesis. The S literally stands for synthesis. So we know that cells spend most of their time in interphase, and like 90% of the time, right? Mitosis or nuclear division is a lot faster because the DNA is kind of exposed there. It's kind of dangerous. The cell lingers too long in that phase, so it's going to go as fast as it can, and it's really, really short and fast and precise. So during interphase, we know in the S phase that DNA is replicating. Well, why must DNA replicate? Well, it's important because if DNA didn't replicate, then when the cell divides, you would only get half of your DNA in each of the new cells. And then if you divide it again, you would get a fourth, and then an eighth, and then a 16th. And at some point, your cells would have no DNA inside of them. We know that DNA is the blueprint and the instructions for what your cell is, who your cell is, what it does, how it does everything it does. So it needs a full set of instructions in every single cell. Therefore, DNA needs to replicate so that when you're dividing and making essentially a new cell, that cell comes with its own set of instructions already. The process of DNA replication is semi-conservative. As you can see here in the steps, it says step 1, the DNA double helix will unwind. So it's usually in a twisty, like spiral staircase form. It's going to unwind so that it's a straight line. And then in step 2, the hydrogen bonds will be broken with DNA helicase. Now, each strand is going to serve as a template. They're not going to only build off of one. They're going to build off of both at the same time. Step 3, the enzyme that's building is called DNA polymerase, and it'll come and bring the base pairs A to T, G to C, and vice versa for both. And then also double check, and then DNA ligase will glue all the pieces together. At the very end, when you look at this DNA molecule, the old DNA molecule was dark blue. The new DNA molecule that was built is light blue. When you look at each of them, you kind of notice that they're not entirely new or entirely old, right? If you just look up the word conservative, it means that you're retaining or holding on to some things, right? And the word semi means half. So when you look at a new strand of DNA that's being built, it's technically half old and half new, right? That's why it's called a semi-conservative process. It's not fully conservative. It's not a fully new strand either. It's semi-conservative because every DNA strand is half of the old strand it was built from. So after interphase, the DNA is replicated and the cell is ready to begin dividing, and it's going to enter mitosis. As a preview, I do want to see how much you remember. Drag the red dot to the cell that you think represents prophase. Drag the green dot to the cell that you think represents telophase. Drag the blue dot to the cell that you think represents anaphase, and then drag the orange dot to the one that you think represents metaphase. So there are 4 phases of mitosis. We remember them by saying PMAT or PMAT because prophase comes first, and that's when the chromosomes are condensing, the nuclear membrane is starting to dissolve because if it stays there, it's going to get in the way. These centrioles are forming their spindle fibers that are going to soon attach to the centromere of the chromosomes so that they can be pulled to the right place and then pulled apart eventually. In metaphase, the M in metaphase also stands for the M in middle. This is where the spindle fibers have moved all of the chromosomes to be in a single file line in the middle of the cell. After metaphase, we have the A for anaphase, which also stands for the A in apart. The chromosomes, or rather, their sister chromatids, are being pulled apart towards opposite ends of the cells by the spindle fibers here. This ensures that every chromosome, where it's already doubled, will then be moved to either cell on opposite ends when the cell is actually splitting. So the cell gets a full set of DNA. The T in PMAT stands for telophase, or for 2 nuclei, because once the chromosomes are pulled to their opposite ends, they start to decondense. And instead of being in that chromosome shape, they're going to go back to their loose, woven spaghetti pile mess. And then the membrane of the nucleus is going to reform around them to then go ahead and protect them again. Again, the PMAT for mitosis stands for prophase, metaphase, anaphase, and telophase. Technically, mitosis is only the nucleus dividing. I want to be very clear about that. It is not related at all to the cell itself physically separating into 2. Okay. Prophase, again, is getting ready to divide. Metaphase, the chromosomes are lined up in the middle. Anaphase, the chromosomes get pulled apart. And then in telophase, there are 2 nuclei that reform. But this is all still happening inside of one single cell. Okay. Mitosis splits the nucleus so that we can make sure each new cell that's going to be formed will get half of the duplicated chromosomes, aka 1 full set. So again, as a refresher to go over what we just talked about, I'm going to ask that you list the 4 phases of mitosis in correct order. Your answer should be PMAT, right? PMAT is really helpful here because it gives you the order of prophase, metaphase, anaphase, and then telophase. Now, cytokinesis happens after mitosis. And cytokinesis is where, now that you have 2 nuclei are going to get split into 2 new cells. The physical separation of the cytoplasm completes cell division in cytokinesis. In cytokinesis, we're dividing the cell. So the cytoplasm will form a cleavage furrow. The simple way to describe it is it just pinches inwards. You can see here in this image that it's going to pinch inwards at the cleavage furrow, and then eventually it'll pinch in so far that it fully separates the 2 cells. And now you have 2 separate cells, each with their own nucleus that we separated during mitosis. Okay, we have images here for you to look at. Which 2 phases of mitosis are being represented by A and B in this microscopic image? These are realistic actual cells, but they are stained so you can actually see them. Human cells tend to, or animal cells tend to be like a neutral color that you can't really see until you put a stain of ink on them. So we have A over here. Is A prophase, metaphase, anaphase, or telophase? We have B over here. Is B prophase, metaphase, anaphase, or telophase? Go ahead and write your responses for me on the pair deck. A is technically anaphase when you look at the actual place that the chromosomes are now in. The chromosomes have been pulled apart to the opposite ends of the cell, A for apart. On the other hand, B is probably metaphase because you can see that the chromosomes are all kind of lined up in a single file line in the center of the cell. Now, is it a neat process? No, but you can kind of tell that it's very different from A, where it's anaphase and they're pulled apart. Now go ahead and drag the icon, which is the red dot, to the appropriate part of the cell cycle that actually splits the nucleus for me. The answer here is going to be the one that, we're going to put the one on the actual part of the cell cycle that splits the cytoplasm on this side. I'll go over the other ones now. So go ahead and put the icon on the part of the cell cycle that splits the cytoplasm. The last one was for the nucleus. This one is for the cytoplasm. They are technically different parts of the cell cycle. Going back to the previous one that I almost gave you the answer to right away, the part of the cell cycle that splits the nucleus is mitosis, and the part of the cell cycle that splits the cytoplasm is cytokinesis. It's aptly named, right? Cytokinesis, cytoplasm. So during the cell cycle, at the very, very, very end of interphase, you enter mitosis, where the nucleus is split. And then at the very end, before you actually restart the cell cycle, that's when cytokinesis happens. And each of the two new cells that have been formed by cytokinesis will then re-enter interphase in the G1 phase. So the age-old question is, what happens if the cell cycle messes up, right? We know that it's good and dandy if it's doing its job, because then your cells are just doubling and doubling and doubling. But the reason why they don't mess up that often is because there are checkpoints during the cell cycle that prevent damaged cells from duplicating. They either will kill off the cell, or they'll, fix the cell up a little bit if it's not too big of a mess up. This happens between G1 and S to check if DNA is ready to replicate. It happens between S and G2 to check if the DNA was successfully replicated. And it happens during the beginning of mitosis to check if chromosomes are aligned properly in the middle so that they are going to get separated equally into each new nucleus. Sometimes these checkpoints don't work either. And you get issues like cancer where the cell just divides uncontrollably and you get tumors that then spread and become cancerous. So it can be really bad if the cell cycle messes up, right? Cancer cells, they completely ignore signals that are telling them to, hey, maybe you don't need to divide. We don't have room for you to divide. Why are you using up this energy and nutrition to divide? Cancer cells just ignore all that. And they keep dividing and dividing and dividing and dividing, even when they're not supposed to. The cool thing is that when you grow normal cells in like a petri dish in a lab, they'll grow until they touch the end of the petri dish, and they'll only grow in a certain number of layers. But cancer cells, they don't stick to the layers rule, right? It's really messy here. You can see in this blue part of the diagram. And they'll grow and pile and crowd out the other cells, even if there's no more space inside of the petri dish. Cancer cells have unregulated cell division, meaning they continuously go through the cell cycle, even if the cell has mistakes. So that's why we worry about skin cancer, for example, when you get sunburns, because the UV can damage your DNA. And then if those cells that have that damage become cancerous, then that damaged DNA is multiplying as the cells are multiplying out of control. Okay, so let's do some practice problems. Again, you're going to make sure that you're reading and highlighting, asking the question in your own words, sharing the knowledge that you have, and then eliminating and choosing the best answer in the final stage of your RAKE method. So this question we're going to do together here says, the diagram shows cells in different phases of mitosis. A student is trying to find a cell in a particular phase of mitosis. The student is looking for evidence that spindle fibers are separating the chromosomes to ensure that each new nucleus has one copy of each chromosome. So we have cells 1, 2, 3, and 4 here. The question here at the bottom says, which cell is in the phase of mitosis that the student is searching for? Well, we got to read and highlight and mark out the important parts. So we're talking about a particular phase of mitosis, and the description of that phase says the spindle fibers are separating the chromosomes. Okay. What is the question actually asking? Well, it's saying which picture is showing the spindle fibers in the process of separating the chromosomes. What does that mean? Well, logically, or if you need a little help, you can glance at the notes sheet. It's saying that you just need to know that the spindle fibers are separating sister chromatid. I don't even need to know what phase that technically is called, but I do know that it is anaphase. So therefore, we can eliminate and choose our best answer. The spindle fibers in 3 and 4 aren't even present. So automatically, you can get rid of them, right? Take a quick guess now which one you think it is. The answer is going to be cell 2 in the very end, because while in cell 1 there are spindle fibers, they haven't separated the chromatids from each other yet. Only in 2 are they actually separating these sister chromatids to the opposite end of the cell, just as the description was asking us for. So now it's your turn to practice. You're going to show your work and explain all of your logic for your answers for questions 1, 2, and 3 here. This whole thing is student paced, so you'll work through at your own speed, and then at the end of the school day, I will be grading it for you. Once you finish these, go ahead and do your exit ticket, and that's all we have for today.